Thanks everybody for coming to CALS for Groundbreaking Discovery in CALS seminar. My name's Tom Zinn and I work here at UW-Madison. I also work for UW Extension Cooperative Extension. Thanks again for coming to the seminar. Later this week on March 15th, the talk will be on zinc metabolism in yeast by David Eide from Nutritional Sciences. A week from today, Sally Leong from Plant Pathology will be here to talk about plant microbe interactions. And then on March 22nd, Greg Gauthier from Medicine will be here to talk about fungal pathogens. And then it's spring break, so you don't have to come. Yay. Today it's my pleasure to introduce to you Roger Sunde. He's was born here in Madison, went to Madison West High School just down the road. Came here to UW-Madison for his undergraduate degree in biochemistry, and then got his master's and PhD here, also in biochemistry. Then he postdoced at the Rowett Institute in Scotland, went on the faculty at the University of Arizona, and then the University of Missouri. And in 2004, he returned to UW-Madison to be on the faculty here. Today he's going to talk with us about dietary minerals. Would you please join me in welcoming Professor Sunde to CALS Groundbreaking Discoveries. Thank you. This is where we clap. Yay! Thank you.
Tom, thanks very much. I'm delighted to be here, and I get to talk about something that's really a bit of a passion with me. So the focus, this whole seminar series is really focused then on groundbreaking research here at UW-Madison. And you guys have heard about Babcock and the Babcock test for fat. The people who've attended this have heard about the single grain experiment with cattle, learned about the discovery of vitamin A and so on. And what I want to talk about now is how that research, really that legacy, gelled into a paradigm for studying essential nutrients. And I'm going to really tell you about a decade from 1923 to 1933 where this really was firmed up, and it became then the prototype for how the essentiality of virtually all the nutrients were discovered. So that's where we're going. So let's look back about 130 years ago in 1988. That was the year that Babcock was actually hired and brought to the university to head up the Ag Experiment Station. And at that time, these three men and these three ideas really dominated what was nutrition. So if we look at this, Liebig in Germany then was really the founder, along with other people, that looked at using proximate analysis to understand what should be in a diet. So people had understood that you needed protein, you needed carbohydrate, you needed a little bit of fat, and you could analyze that. You could throw in some ether and extract the fat out and determine how much was there. You could digest that and determine the amount of nitrogen, and from that you could calculate then the amount of protein that was in a diet, and except for the ash, what was left then was carbohydrate. And so that was the idea about how you could determine whether a diet was good or not, how much protein, carbohydrate, and fat was in the diet. At the same time, Louis Pasteur then had proved the germ theory of disease. So disease wasn't caused by bad air. It was caused then by germs. That was the major thinking. Diet was thought not to be involved. And lastly then, Atwater had worked out to calculate then the amount of carbohydrate, the calories in carbohydrate, the calories in fat, the calories in protein. And this was a way then you could assess the best diet that was possible. So Babcock, our star then, wondered about this. And he said, what if it mattered more than just proximate analysis to determine that? So as I said, Babcock came here in 1888, and 2 years later he developed the Babcock test to determine the amount of fat in milk. So farmers could no longer water down the milk and then sell it by volume. And this was quite important then under these conditions, and it made Wisconsin the dairy state. But Babcock had these strange ideas then about maybe it mattered more than just proximate analysis with regard to nutrition. And so one of his first steps was to hire somebody by the name of Hart, Edwin Hart here, who came then and was the first chair in biochemistry. Now, before I get started, he also hired McCullum, and McCullum is the one who discovered vitamin A. And I want to mention that. So McCullum then was brought in, along with Hart, to really do the single grain experiment. And so there were 4 groups of calves, as you remember, and they were formulated with balanced diets that came from a single grain. So all, the chaff, the protein, and so on would all provide the right amount of carbohydrate, fat, and other nutrients, and there was either based solely on wheat, solely on oats, solely on corn, or on a mixture. And the point is, the ones that were fed wheat soon died, and some died, and their calves were stillborn, and so on. So McCullum was the guy who had to mix these diets. And he's doing these experiments, not on mice, but on cows. There's a lot of work to mix those diets and so on, and he began wondering about what if he used small rodents. So he first went down to the dairy barn and trapped some rats and tried to do studies with that. And that wasn't successful. So he actually went down to Milwaukee and for $6 bought some white rats and brought them back to Madison. And that was the founding strain of rats, not only for what's done in biochemistry here, but also for many of the rat strains that are used throughout the United States and the rest of the world. So with using rats, he did an experiment. And I'm going to roll up my sleeves experimentally, and we're going to look to see the nature of how data was presented at this time. So this is a JBC article. I want you scientists to notice how critical statistics are going to be in this report. So what they did is they fed rats then a diet, supplemented with lard and minerals, and they let them grow for 4 to 18 weeks, okay? And their growth would slow. And you can see this is a normal growth rate, grams here, and this is weeks here, okay? And if they just let them grow like this. But then if they could restore growth, if they fed them some egg, or if they ether extracted the egg, so something that's organic, that would allow them to grow. So here's the ether extract, and they suddenly would start to grow. And you can see right here, this female rat got pregnant, and then she had a litter. You get all this little information back from JBC in those conditions. Or they could feed butterfat, but olive oil or cottonseed oil wouldn't. But if they extracted the butterfat and then shook it with the olive oil, then that extract mixed with olive oil would restore growth. And they called this substance vitamin A or something like that. So again, we're seeing the beginnings of this where they're extracting a foodstuff and putting it back into the diet and see if it can correct or improve growth or health. So where are we right now? This is a picture, I think it's really a postcard rendition of what the biochemistry department, then agricultural chemistry, so here's what faces Henry Mall. This was the 1917 wing. Just the front of that is there now. And the 37 wing was back in the bed. I include this here. This is me in a younger, more hairy age. So I grew up in Madison. More importantly, my father then was a student of Elvium's, and he actually then collaborated with Elvium later on. It turns out he was his pallbearer as well. So I heard all kinds of stories about this kind of thing at the time, and he interacted with Hart and Elvium and Steinbach as well under those conditions. And I obviously was in the building that goes there, just underneath the rat room, and a lot of the apparati were still there. So I'm going to tell you a few of those stories. I'm not going to tell you the good ones, but I'll tell you a few of those stories right now. So in 1923, this was the list of essential nutrients. So we know calcium chloride, iron, phosphorus, potassium, sodium, and iodine, and then obviously nitrogen and sulfur. They were required for living organisms under those times. So that's the situation. So this is an excerpt that was written by James Waddell, and I'll tell you more about him. And he was a student then of Hart's at the time and received his PhD later. And he described this some 60 years ago. But so Hart, Professor Hart then, was interested in mineral metabolism, and he was particularly interested in iron and also interested in milk. And at the time, people knew that iron alone would not necessarily correct anemia. There was a missing piece there, and so he began to want to explore that under those conditions. So let's set the stage. So Edwin Hart then was brought in as the first professor and chair of agricultural chemistry. He came here in 1906, and he was chair until 1944. He died in 53 then, so he continued to work after that as a retiree. And he had worked on the phosphorus requirement of proteins. He developed an assay for casein, the protein in milk as well, using the same centrifuge actually that Babcock used. And that was important, it's really important as we formulate and price milk today, assaying protein as well. And he also, because there was money being available, he outlined then a series of projects to look at the requirements for minerals and vitamins. He wanted to use animal models, he wanted to use plant models, and he wanted to use microbes as well. And all three of those lines of research continued then through much of the last century in the biochemistry department. So this is the first JBC paper, okay, in this series, paper number 1. It was submitted in 1925. So here's Professor Hart, Steinbach, the young faculty member, or young assistant, Elvium, even younger, and Waddell, even younger than that, on this paper. The nutritional anemia of whole milk diets and the utilization of inorganic iron in hemoglobin building. So this is the stage from which we start. And people knew at that time about hemoglobin. So here's iron centered in heme with the porphyrin rings. Okay, very important for red cells. But their hypothesis was this other factor was likely to be an organic factor. Remember, this is done right after we've discovered vitamin A here on campus. And so they postulated, for instance, maybe chlorophyll was necessary, very similar to heme. Maybe that was what was necessary. Or they postulated maybe vitamin E. So Evans had discovered that vitamin E was a vitamin and important in reproduction in small animals, rabbits, and other rodents. And so they were really aimed at identifying an organic factor that was necessary for the assimilation of iron. So let's take a look now in some detail at this experiment. So this is how JBC reported the data at the time. One animal at a time. There's rabbit 1, 2, 3, and 4. And they tell you something about the diet. So they fed them, this is the dairy state, they just fed them milk. Or they fed them milk and they put something in. So this is fresh milk hauled down in cans from the dairy, the biochem dairy barn down to biochemistry, and they supplemented that. And what you can see here is that if they were just giving milk at all, the hemoglobin levels dropped, the erythrocyte count dropped, and they didn't grow quite as well as otherwise. And I've plotted that on a scale here, so you can see this pair of unsupplemented ones, and then the ones that received cabbage. Okay, and they obviously grew quite a bit better. Their erythrocyte counts didn't drop. Okay, and hemoglobin stayed about the same as well. So there was something in cabbage, that organic factor perhaps, that might protect. So let's look at the bottom of that chart. So here, if they put iron in there alone, the hemoglobin still dropped and they didn't grow as well. But if they put milk plus iron plus cabbage or dried cabbage, they also then maintained their hemoglobin levels, they grew, and so on. There's a panel of research like what one would see in JBC today. So what did they find? So they tried fresh and they tried dried cabbage. That contained some iron. So maybe it was the iron in that product that was really allowing, protecting against the animals becoming anemic. They did alcoholic extracts then of cabbage or cornmeal, and this would protect, plus iron would protect alone, and these extracts were free of iron. So it was something else. Purified chlorophyll. So note this as well. They didn't order this from Sigma. They had to purify chlorophyll then, add it every single day, a couple drops to the bowls with various things, and see, and that would also protect. So this is sort of a summary of where we were in 1925. Rabbits then were limited to a whole milk diet. They developed anemia. Inorganic iron wouldn't correct this, but fresh or alcoholic extracts of cabbage and so on, and chlorophyll would all correct the anemia. So that was in 1925. So now I'll tell you just a little bit about Harry Steinbach. So he was a Madison native, or excuse me, a Wisconsin native as well, a farm boy, grew up in New Holstein, I think. And he was actually a co-author. So he was one of the single grain experiment as well, along with Elvium and Hart and so on. Babcock wasn't an author on that. He was just the ideas guy. He received his PhD in 1916, and just about this time then he discovered if you could UV irradiate food as well as animals, and that would protect against rickets. And this also led to the patent in Wharf, and I think we had a speaker earlier in this series that talked a little bit about that. So Harry Steinbach was one of the people. And so one of the key things I want to emphasize now is that stock diet, for instance, was used in showing the irradiation of food or showing the vitamin A requirement. And so he published a paper. Again, this was published in Science under those conditions. And so what he did is he had a mixture of corn and linseed oil and casein and alfalfa and a few salts, plus fresh milk, and that was their animal model at the time. But he thought it was important to publish that, and it was of sufficient quality that it needed to be in Science. So this was the adequate diet at the time. And so the anemic rat model then, publication 5. So there's 3 other publications in between. This is just 3 years later. Okay, and I've sort of sketched out, you've got a normal animal and an anemic animal, and we've still got Waddell now as the first author, Steinbach, Elvium, and Hart under those conditions. Okay, so again, they're still presenting in JBC. Here's rats, individual numbers, erythrocyte counts, hemoglobin counts. And so if they just were fed the whole milk diet, they would develop anemia. But the whole point is that they could now analyze for hemoglobin, and this is actually data I pulled from another accompanying paper, but you can see the progressive decline in hemoglobin levels in animals just on the milk diet. And this actually is an average then of something between 22 and 97 animals. So they had a biomarker for doing their assessment. They couldn't just follow weight and try to, and this really helped focus their attention, something we all know today is important. So James Waddell then, in that review some 60 years ago, talked about that. And so our weanling rats restricted to a milk diet developed a profound anemia in a few weeks, and this was highly sensitive then. So then they could start putting supplements back in and see if that could correct or protect against anemia. This is the biological method of analysis. It's completely different than from Atwater and Liebig, who were suggesting you could do proximate analysis, chemical analysis, to assess the quality of a diet. And so that's the key thing that really arose out of this period here at the University of Wisconsin. So the question was, remember the purified chlorophyll plus iron, so maybe that's an organic factor. So how can we attack that as well? So I'll tell you just a little bit about James Waddell. So he got a PhD from Hart in 1926. Interestingly, he went off to DuPont then and worked there the rest of his career in industry as well. He directed research. He provided a lot of amino acids and vitamins. And if you scale through the 40s and 50s and 60s, you can see thank yous where they've donated lysine or methionine or something like that, provided that to researchers at the time so they could do their experiments. And he studied iron fortification products, iron that might be put into bread, for instance, to help provide extra iron. And he actually then applied for and succeeded in getting a patent for the synthesis of the anti-rachitic factor, that's vitamin D, or ergocalciferol, making that from cholesterol then. And again, important in industry at the time. This isn't him, that's his son. I couldn't find a picture of him. So here's publication 6, 3 years later, okay, under those conditions. Okay, received on March 17th. I'll come back to that in just a little. Iron salts and iron-containing ash extracts. So now let's see if we can ash it and separate out the organic from the inorganic and see if that's what has this anti-anemia factor that's needed in addition to iron. Waddell, Elvium, Steinbach, and Hartman under those conditions. We're still back reporting individual rat weights and individual rat hemoglobins. Okay, and you can see these are rats that on their diet, just given iron alone. Here's 1 milligram of iron, and there's little crosses here saying that after each one of these lines is 6 weeks, that animal died. Okay, So that's the control. And here are animals then that are given ash of dried lettuce would promote growth. Okay, you can see the growth is good and their anemia levels, hemoglobin levels are also good. So the protective factor has to be inorganic then because they've ashed it in all their feeding. And again, what are they doing? A couple mils of milk every day. One of the students, probably Waddell, went down and probably used a mortar and pestle and blended a little bit of this ash into the milk that particular animal. I think they actually fed them in mortars from a mortar and pestle then, put that into the cage then to feed the animals. So maybe it was the form of the iron that was important. Maybe there was a different form. So they synthesized then ferric sulfate and ferric acetate and citrate and phosphate and chloride. And what you can see is in each case, it didn't promote hemoglobin levels. So it wasn't a particular form of inorganic iron that was protective. There was something else under these conditions. Okay, so in that same paper then in 1928, maybe the source of the factor is important. So they provided dried beef liver, kidney, muscle, corn, and wheat, and you can see individual animals in this plot here. The dried liver would promote hemoglobin levels, but from kidney, from corn, from wheat, from muscle, that wouldn't do it. And they also added dried foodstuffs where they analyzed for the iron, so they were providing iron as well. So there was a lot more of that substance. They ground that into the milk as well. And it still, except for liver, didn't protect. So my father actually was still kicking around as a grad student when Hart was around, and he actually sent him down to a store, which would be sort of under where Union South was, to get some tomatoes to dry and feed in some of his later experiments. So this kind of thing was going on all the time. Okay, so it doesn't look like these other products would provide it. So ash lettuce was just as good as lettuce. Inorganic salts didn't fix this. Beef would really not correct this anemia. Okay, the 0.5 milligrams of iron in dried liver would reverse it, but smaller amounts would not. So they said that these ashes and extracts, in addition to iron, contain other inorganic substances that are vitally concerned with the building of hemoglobin. So they really had the idea framed, they just didn't know what to do. Okay, so let's take a look here. This is the publication, March 23rd. This is 6 days later. This is issue 7. So I don't think they did all this work in those 6 days. This isn't, like making earth or something according to the Bible. But again, Hart, Steinbach, Waddell now got kicked down a little bit. And the title was Copper as a Supplement to Iron for Hemoglobin Building in the Rat. Okay. So at the time, pernicious anemia was a real issue. This was the AIDS of the early part of the 1900s. If somebody got pernicious anemia, you were slowly over time going to die. And there was nothing that they could do to protect against it. People didn't understand it. But people did discover that there were factors then in liver that might protect. And so drug companies, again, people had liver pills and all kinds of other things, but okay. Eli Lilly had a particular preparation, okay, which was very effective in the treatment of pernicious anemia, and it was low in iron. So this cooperation, that's where I really want to come from here. This cooperation between industry and the university was very important back, and it wasn't thought to be a bad thing. It was thought to be a good thing under those conditions. So what are they going to do? Here they can provide the Lilly preparation with that has a small amount of iron, and it didn't protect. So the animals still grew, but their hemoglobin levels were repressed. But they could provide the Lilly prep plus iron, and their hemoglobin levels darted right back up, and their growth grew as well. So what are they going to do? Let's ash that preparation and see if it still protects or not. Is there some organic factor still in this preparation that's allowing for the assimilation of iron and incorporation into hemoglobin? And so again, we're still reporting single animals. So here's the ash plus iron, and in each case, hemoglobin levels are getting up over 12, you can see, and growth was pretty good. Okay, so let's take a look. Let's do an acid digest of the lily preparation. Really chew up that organic factor. It still promoted growth as well. Okay, so they could also do a hydrochloric extract under those conditions. Okay, so this is just the soluble phase, and that still would protect, but the residue from that HCl extraction didn't protect. You can see that there's crosses here. The animals died and they didn't grow, or their hemoglobin levels stayed very low as well. So there was obviously something then that they could extract with iron, or extract from the Eli Lilly preparation. Now this had a faint blue color. Faint blue color. Anybody here take analytical chemistry back in the days, or okay, when you had to determine what was in an inorganic mixture? One of the steps then was people would bubble sulfuric acid, smells like rotten eggs, through that mixture, and they would precipitate cadmium, they'd precipitate copper out of that. They were black little precipitates. Okay? So they looked at the same thing, and the hydrogen sulfide fraction then resolubilized. It promoted growth, but the precipitate from that fraction, okay, this was the only potent fraction, so the soluble phase was not. There was something then that was precipitatable by hydrogen sulfide. It could have been cadmium, couldn't it? But the blue color probably suggested something else. This is the microarray of 1928. So what did they decide to do? I'm quoting, "We made this trial of copper salts because it fitted in our scheme of testing all the inorganic elements, the supplements to iron, okay, which had been recognized as present in biological things. So we'll just test each one. A little bit of milk, grind in a little bit of this and see whether it helped or not." Huh. It took, how many years did it take them to figure that out instead of doing all this other work? Okay, so under those conditions, and lo and behold, here's rat 621. This is our star then. Our first attempt in correcting the anemia, okay, so they added 0.5 milligrams of iron and 0.25 milligrams of chloride. And you can see, here's the growth of that rat in the anemia, okay, about 12 weeks in, 13 weeks. Then they provided the supplement, I guess it's 4 weeks in this grid, and lo and behold, hemoglobin levels went. So without the copper addition, the rise in hemoglobin would not. So this is the, rat 621 is the first demonstration of the essentiality of copper. Again, the biological method of analysis, have a diet that's crummy, develops disease, and add individual components back, focusing down until you can find what supports growth. So this is the full page for that report. And so they said, we think that this is the first experiment in the literature giving to copper an association of iron that's specific for hemoglobin building in a mammal on an otherwise satisfactory diet 90 years ago. So there's the story. Okay? So this liver preparation was really important, and so they've indicated now we've secured no evidence for the existence of an organic factor, all this extracting, all this ashing that's necessary for hemoglobin synthesis for animals kept on a whole milk diet and supplemented with inorganic iron. And so we might advance the orthodox suggestion that copper acts as a catalyzer for some reactions concerning hemoglobin building. Again, they always sort of soft pedaled some of their results a little bit under those conditions. So I'm going to shift gears now and come back to Conrad Elvium. So Elvium used to have his name on the Art Center. Now Chazen, who's donated more money, I guess, what did he get his money from? Claiborne, I think, something like that. So they kicked Elvium's name off the Art Museum now. But so he got his PhD with Hart in 1927, and he was responsible for unraveling this complex of the B-soluble vitamins. So B1 and B2 are very similar. One of them was heat stable, B1, thiamine, which cured beriberi or a neuritis in pigeons. Notice I've been talking about a whole series of different kinds of animals under these conditions. It isn't just rats, and it isn't just cows and pigs and so on under those conditions. riboflavin in yeast, that's not heat stable, so they were able to separate those two factors under those conditions. But he worked with a wide variety of animals. His main focus was the chick, and he and my dad published a lot of papers. So in 1937, they purified nicotinic acid, and they showed then that this was what was missing in Pellagra under those some years later on. So when Hart resigned in 1944, Elvium became chair of biochemistry, and he was chair until 1958, and simultaneously he was dean of the graduate school until 1958, and he became president of the university in 1958, and he died at his desk in 1962. So Elvium, but the big thing that he did in addition to these other papers was really unraveling the various vitamin Bs 1 through 6, roughly, under those conditions. So in the intervening time between the demonstration that copper was essential and what I'm going to tell you about manganese here at the university, Kemmerer, Elvium, and Hart showed that manganese was an essential nutrient. And McCullum, he left in 1917 then, and was at Johns Hopkins, and they showed that magnesium was also an essential mineral. Okay, so I'm going to tell you now, Wilbert Todd, got his PhD with Hart in 1933, so he's the first author on the zinc work I'm going to tell you about. And he became a professor at Oregon, the University of Oregon, and he did a lot of research in sugar metabolism, carbohydrate metabolism, and he was, along with West, was author of a textbook of biochemistry, went through many editions, and was a leading text into the 60s and 70s, actually. And this is a picture of him working with a student. So this is the key paper, published in 1933. It actually was submitted in September of 33, and because of that particular volume spanning into 1934, many publications say that the work was really done in 1934, but it was 1933. And so the focus of their work then was to see, using this biological method of analysis, is zinc does it have a beneficial role or not? So one of the things is you've got to be able to design a diet that's deficient in the nutrient if you're going to use the biological method of analysis. So here's a paper, again, Todd and Elvium in 32, and this just shows a series of extractions and other things that were used to focus in so they could finally get to a colored zinc compound that they could then determine the quantity of zinc in diet materials to formulate a deficient diet. Okay, so in 1933, they developed then a complete diet. They discovered that distilled water from the tap was not sufficiently free of minerals under those conditions. And this is their diet. This is virtually the same diet that's used today. There are a few exceptions, but when I was in school, people still talked about these two salt mixes that provided the minerals that I talked about and had been discovered in the intervening time. So here's manganese, here's magnesium, copper, iron, calcium, and so on. They provided berry, B1, thiamine, and rice polishings. Egg white contains riboflavin, so they provided that, and that would have been cooked, I think, so that some of the other agents there didn't interfere. They provided A and D then, which they knew to be essential, and some oil as well. Provided 1 to 5 drops daily. So Todd is probably in there grabbing one of these friendly rats and dropping a drop or two of these materials into their mouth every single day. So here's the growth. We're still reporting. This is the American Journal of Physiology. Individual growth. Here's animals on a zinc deficient diet, 6-week intervals here, and you can see they're getting up to be just a little over 100 pounds or 100 grams. And you can see these are the animals that got 50 parts per million zinc in addition. A clear demonstration that zinc would promote growth in the animals. In addition, what they noticed was the animals that were on the control diet without any zinc began losing their hair. And so a photograph in this article then shows the underside of this female rat, 17 weeks on the deficient diet. And after 7 weeks, the bare spots were almost completely recovered if they then switched them back to a zinc supplemented diet. And so this paper then demonstrated that the zinc deficient diet resulted in an inferior poor growth. 50 parts per million zinc, that's a little bit higher than what we think the zinc requirement is in the rat today, was effective, and this was involved in hair. And so they went on to say it seems evident that the rat ration was not complete. So, and so there might be additional factors that aren't present in water-soluble B that are necessary. And they went on to say there might be the possibility that a lack of further inorganic elements, such as aluminum or boron or arsenic or others, might be important. Okay, so that was 85 years ago in this decade, showing the biological method of analysis and what tremendous progress had been made. And you can see other people then picked this up to establish the essentiality of other nutrients. So in red is this little period that I've shown you here. And then soon after, cobalt was shown to be essential in a ruminant. Okay, there was reports that fluorine or fluoride was essential. Molybdenum, more because of its need for sulfide oxidase. And selenium was discovered in 1957. But you can see there's a list of other elements that many people think are required for higher animals that really haven't been discovered, haven't been characterized at this time. Now, Dr. Eide is going to tell you the punchline, but I thought I would just point out, for instance, we now know that copper is involved in some oxidases that are necessary to get iron in the right oxidation state so it can be absorbed. We now know that if individuals, humans, are missing a particular copper transporter, it results in an inherited disease, Menkes disease, under those conditions. Zinc is similarly involved in zinc fingers, okay, chelated by cysteines or cysteines and histidines. That they're like the fingers that are so important in identifying or regulating gene expression. And this is from, oh, come on, Roger. This just shows a whole series of different steroid receptors, vitamin D receptor, androgen receptors, all having zinc fingers. And this illustrates then somebody without zinc, and there's profound developmental problems, reproductive problems, and so on under those conditions. And again, if there's a zinc transporter is missing in humans, it results in a zinc deficiency disease. So I want to finish by saying, what has stopped this progression? This wonderful biological method of analysis, why haven't we chipped away to demonstrate that boron is essential for humans, or aluminum, or maybe even chromium, though I don't believe chromium is essential. So what's going on there? One of the things that is pretty clear is maybe we can't make diets that are sufficiently deficient, though people have worked with going after the ultra trace elements under these conditions, and this has not really been successful. I think one needs to realize that organisms have homeostatic mechanisms that will really hang on to and preserve nutrients to the point that it sometimes takes multiple generations to make animals deficient. And there may be overlapping roles then. So if you're missing one nutrient, there are other enzymes, other processes, maybe not requiring minerals at all, that might do the same thing under those conditions. And there may be some minor pathways that might provide a little bit of alternative synthesis or something. But really, we've got a lack of good models for deficiency for many of these things under these conditions. We don't have a good biomarker to show whether or not, what happens if you don't have enough boron? What happens if you don't have enough nickel? Okay, we're missing those biomarkers to really demonstrate that. And it may also be that this is only important in certain stages of life, and it's pretty hard to test what the elderly human needs in a rapidly growing rat under those conditions. And there may be rare conditions under those conditions. So what I wanted to do today was just show you some of the actual data from that decade so that you could get an idea about what they did. And I wanted to sort of play around a little bit to show you that there wasn't a lot of statistics, but certainly people believed it, and this has really established where we are today. That all arose then out of Babcock, Hart, McCullum, Steinbach, Elvium, and then a couple of young assistants that went off to fame and fortune other places here. But this was really the cradle of establishing the biological method of analysis and identifying these essential nutrients then under those conditions. I also want to point out the use of multiple species. Obviously cattle, hogs and chicks were involved, though I didn't talk about them today. I mentioned pigeons, rats and mice. A whole series of animals have been used. There's nothing wrong with studying human nutritional biochemistry in these other animal models as well. And I think that's something we need to remember also. So I'll stop there and see if there's any questions. Thank you for coming and your kind attention. I guess I can see if there's any questions. There may not be.
I've never seen the 1917 edition talked about in the biochemistry building. Do you have?
I actually pulled that from the biochemistry history piece, and I was startled by that postcard-like graphic representation.
I've seen that a picture like that before, but I've never seen two dates on it, and I don't know that it's in history that Jim.
No, I was quite confused about that as well, but I was trying to understand that, and I'm not sure if that was part of the 1937 edition or it was a separate edition. But I actually, in the history of the biochemistry department, I actually saw that postcard. And I was trying to get as close to this era as I possibly could. So I don't know whether there are some interesting structural, having been a student there's some interesting structural differences in that gap. That's where KP Link did a lot of his work in that intervening little segment. But
along those lines, the 1937-38 edition that you mentioned, my memory was there used to be a plaque up there that said these are dedicated to the Babcock laboratories.
Yeah.
And that plaque is missing. I think they took that plaque, and it was in the west wall.
Sure. Yep.
Has Dave Nelson talked to you about that?
No, he hasn't talked to me, but I recall seeing that years ago. Yeah.
If you can find a photo of that, that would be very interesting because of the whole issue of what that name of that building is.
Sure. Yeah.
Because Babcock died in 34, 33, 35, and that would have been the first.
Somewhere around there. I forget exactly when now. It was interesting. He was studying gravity in his later years. I think people start doing strange things later on. So he had all kinds of apparati in the basement of the biochemistry, and he was studying gravity at that time. And I'm not sure what he discovered, but nonetheless. Guy.
The first is that you said agricultural chemistry, so biochemistry used to be agricultural chemistry?
Sure. If you look on the lintel going in from Henry Mall, and they may have covered it up now, but it used to be etched in that agricultural chemistry. So originally it was agricultural chemistry. It wasn't bio.
Where I'm sitting right here, it says agricultural chemistry.
Yeah.
So the other question is, so these things, the minerals and the vitamins are being discovered based on growth rates, largely, and extractions, etc. At what point did the I guess it would be enzyme function, the cellular or biochemical function would start to come out? Was it going on simultaneously?
Yes, in many cases it was going on at the same time. Sometimes it actually preceded the demonstration of nutritional essentiality. It's pretty, if you have an example, so people knew about carbonic anhydrase and zinc, for instance. I think that's right, Dave, about that time. I think that carbonic anhydrase was crystallized back in the 30s, and they knew it contained zinc, for instance. So that those things went on. We're in situations right now where Molybdenum is a good example, where it's almost impossible without some tricks to show that an animal requires molybdenum. The homeostasis is so critical, but you can, show that there are molybdenum-centered enzymes, and obviously they need, if you knock those enzymes out, they cause problems, so it's pretty clear that one needs molybdenum. Or you can feed tungsten, which sits right underneath molybdenum in the periodic table, and with a high tungsten diet that doesn't allow formation of the molybdenum cofactor. You don't have to take that as a vitamin. We can make it just like we can make ATP, but the tungsten molybdenum containing molybdenum cofactor doesn't catalyze the reactions like sulfide oxidase. And so therefore you can show, it's pretty clear, even though it's hard to show nutritionally, it's biochemically essential or genetically essential.
So were these discoveries sort of the nature of how the field of enzymology started to take off, or was it, so in other words, you have vitamin whatever, and it's a cofactor for an enzyme in this reaction, and all of a sudden, it seems like the period after this was when all the enzymology started to...
So actually, because of brewing and other things, the enzymology preceded this to some extent. So people had worked out some of the pathways involved in yeast metabolism, for instance, and things like that. So they knew quite a bit about, they knew one of the factors is a yellow factor that's important in metabolizing carbohydrate. Probably has riboflavin in it, I think. But they didn't know anything about exactly what that factor was. And another example would be the niacin, nicotinic acid, and pellagra. That all went, people had figured out there were some diets that might protect, but to really sort that out, somebody had to really purify nicotinic amide and show that that was the key factor in that. So I think it's a blending. It isn't just isolated in one or the other, but from a diet health point of view, these were the key experiments. I mean, it took what, 80 years? No, 50 years to really understand molecular rules for zinc-like and zinc fingers or something like that. So, yeah. But they're going on side by side. That's the way I'd like to think about it. Good question. Yeah.
I also noted that you were very specific in saying that McCollum went to Milwaukee and not Chicago, and to buy the first round of rats.
Did I say Milwaukee?
Yeah.
Oh, I didn't mean to say. He definitely went to Chicago from everything I've read multiple places.
Because I was wondering, when I heard that, I was like, whoa.
No, he definitely went to Chicago, and he had to experiment with, several histories. Schneider has written a really outstanding history talking about rats' fats and the history of nutrition here, and has outlined, but his senior colleagues weren't supportive of using rats under those conditions. He actually went back to the literature then and read about this. And back in the old days, if you went to the Steinbach library, in the back there was a little card that somebody signed, and people have made note that he was probably the only one that ever checked out these journals and read them. So there were lots of people prior to the McCollum era, the Steinbach diets, that were trying to use small animals as models for this, and they weren't successful. They weren't mixing the right ingredients. So the use of milk was probably really an important one that provided many of the essential nutrients that were missing if you just tried to formulate a diet from grains and other things like that.
You made a point that many animals were used, and yet the rat seems to rise to the top, not the pigeon, not the mouse, not the hog. What was it about the rat, even though I don't know much about the genetics of rats?
I think at the time, rats grow quite a bit more. So it's a lot easier to make a deficient rat. So you bring in a rat and they're about 50 grams at weaning, and they'll grow to 250 grams in 4 weeks. You get a mouse in at 21 days and it just inches up a little bit. So by dilution, You know, if you think about it, there's a 5-fold dilution of nutrient in the body under those conditions. And the tissues are bigger as well. So I think that's probably why the rat was the predominant organism at the time. Now mice are sort of a central thing, and we've got the tools to go after that a little bit better than in the rat. But I think that's the reason. And of course, the amount of diet that you mix. I can tell you personally what a difference it is if you're going to do an experiment, let's say with a turkey or a chicken, okay, and feed them for 4 weeks worth versus rats. One needs one hell of a lot more food to do a chicken experiment or turkey experiment than you need for a rat experiment. So just cost and ease. McCullum really had something. The story is that I'm not sure why he was irritated, we'll use that word. But the story is before he left to go to Johns Hopkins, he went up into the attic of biochemistry and he let all the rats out, just to confound their studies. There's a lot of personality that blends into this as well under those conditions.
That doesn't happen in science any
No.
longer.
Dave?
That doesn't.
So Roger, you've really told us some very fascinating history here. It must have been a paradigm shift at the time.
Yeah.
Do you have a sense of how well accepted these ideas of inorganic nutrients were in the field?
Well, I think yes. In fact, a guy by the name of Thomas Kuhn wrote a book in the 60s called The Scientific Revolution, and he cited Babcock and the biological method of analysis, and he talked about Einstein and relativity as his 2 examples of paradigm shifts between, in our case, classical, or in Einstein's classical physics versus relative physics, and in our case, the chemical method of analysis and the biological method of analysis. And it transformed, literally, the study of biology. And it's well accepted, I think. This isn't...
But was there much resistance among colleagues to accepting these
No.
ideas?
People just piled on board. I mean, you can just see the plethora of studies using this. And again, you saw there were a couple of papers where they explicitly talked about the animal or the animal diet. And the diets that you could still buy when I was a student, those primitive salt mixes from 1933. So it was, and again, and I've been at other institutions, that period is talked with some reverence amongst the nutritionists, yeah. Yeah.
So the Eli Lilly supplement from their company, how did they come up with that solution as a treatment for pernicious anemia?
Well, they would have, I think people realized, we still had to eat liver once a week when I was a kid, okay? And so people knew that was rich in whatever was protecting people. So they did extracts and other things, and just like people were doing here. That was, this was a time when people traveled around with bottles of elixir that was going to protect people against. Sounds a little bit like what's going on today, doesn't it? Probiotics and everything else. So let's find something that really nobody knows anything about and see if we can make a profit. But Eli Lilly was well known for that. So they had a collection. They had several divisions then, and they took over other companies as well, DuPont, Eli Lilly, and so on, to make these fractions and then sell them. So Waddell actually published in the 70s something that was funded by several federal agencies and so on, talking about different compounds and enriching flour and other things with iron compounds. What was the best iron compound to put that might take care of iron deficiency in the U.S.? So those companies, and there was money to be made, and they were using science. I think the striking thing is, since 1950 or so, we really haven't found a profound essential nutrient since then. There was a group that was doing some work for a little bit, thinking that pyrrolonequinone, PQQ, might be a new vitamin. And it turned out it wasn't a new vitamin. It was just sort of a funny animal model at the time. But nobody's really... Threonine was, I think, purified in 1947, and that's about the last of these common nutrients that we really... Again, we're not talking about health throughout the lifespan. We're not talking about something in the far, we're talking about the things that really protect against acute disease. The other factors that are really troubling today, what do we do about cardiovascular health? What do we do about diabetes? What do we do about some inborn errors and other things like this? Gluten-free diets. Those are other issues that this particular model alone isn't going to solve. Tom?
I was struck by your analogy of pernicious anemia to be the AIDS of its time.
Yeah.
When I read the plaque across the street, across Henry Mall, it does not use that phrase, pernicious anemia. It just calls it copper-dependent iron deficiency anemia.
So pernicious anemia is really missing vitamin B12, okay, which we can't synthesize from cobalt and the other components, and plants don't make it. So you have to either eat some fecal material or animal products in order to get your vitamin B12.
So that's a different...
That's a different anemia. Pernicious anemia is not the copper and iron anemia.
Okay.
And there are other anemias as well that can be developed, yeah.
Thanks for the clarification.
Sure.
I have a question. I
Yeah.
noticed that multivitamin supplements in the market have some differences in their composition. How can the consumers know which one to take?
So the question is about taking a multivitamin.
Correct.
And I think it is thought if you eat a healthy diet that's a lot of variety, you shouldn't have to take in a vitamin pill, with extra minerals and things like this. Probably the most worrisome is the people that think that they're going to select certain nutrients to take and not others. And there's good evidence to show, for instance, if you ingest too much zinc, you might impair copper absorption. And in fact, the people that have acrodermatitis enteropathica, and they cure them by giving them extra zinc, women, especially of childbearing age, who are they have to give them copper as well, or they may become copper deficient. So the idea is that, if you're really worried, I would take a good multivitamin that's got 1X, a little bit extra of everything, and that'll make you feel protected. But the idea of taking just some at high concentrations probably is not recommended, because at some point these things become toxic, and sometimes they interfere one way or another with metabolism at other areas. So young people taking, another example is just a few years ago it was recommended that the elderly should take vitamin D and calcium. And this is like within the decade. And yet now the data doesn't support and suggests that maybe that extra calcium and vitamin D really isn't helping the elderly. It's not really attacking the problem that they thought they were going to cure. And so we've still got a lot to learn. But I think if you feel compelled, take a good 1X multiple vitamin. Stay away from the vitamin waters. These multicolored things that occupy half a row in the supermarket. They've got God knows what in them, at what levels, and so on. There's no evidence at all that any of those things are going to help you. But they can, if you got money in those companies, you're going to make some money at it. So vitamin water is probably not the thing to take. So my recommendations, in spite of all this, is eat moderately, eat a variety of things, get some exercise, avoid stress. I think if you look at all these things, those 4 things are probably the most important if you want to, from a nutritional point of view, do well for yourself. So that's my recommendation. Tom?
The last major mineral I can think of that you didn't mention is iodine. Is that just a story for another day? Or?
That's probably the story for another day.
Okay.
So the iodine to protect against goiter was actually shown in school girls in Ohio by David Marin in 1917. So that predated the work that was done here in Madison. But the important work here was showing that iodine would protect animals against the same disease, and then the advocacy of iodized salt, those two things were what really made Wisconsin's contribution important. But the demonstration that iodine itself would protect against goiter was done a few years before this. And so if you saw on my little list, it was up there with 1917. So, yeah. I mean, but Madison's fingerprints are all over these other discoveries if you look at them a little bit carefully. So, good question.
Any other questions?
Well, thanks for your attention. I'm delighted to tell you. Ask me sometime and I'll tell you some other nasty stories about this.
I want to know why the B vitamins are named the B vitamins. I heard because they're in beer.
No, I think they had vitamin A, but berry. Actually, one of the reasons was because it was curing berry.
Aren't they the water soluble?
They're the water-soluble B vitamins, yep.
My workers don't.
Good.